So my name is Jim Davis. I've been working on the BV-BRC project since its inception in 2019, but I've been with the Patrick Group since 2013 and a collaborator for several years prior to that. I'm going to show you how to use probably our most useful app service on the website. It's called the Comprehensive Genome Analysis Service. The Comprehensive Genome Analysis Service works by doing a de novo assembly followed by an annotation run. And so what I'll do is we'll launch the assembly, walk through the assembly results, and then walk through the annotation results. So I'm going to start, I guess, with two different tabs going, and I'll start from the home page on each. So in the one, I'm going to traverse to the BV-BRC workshop directory. We keep going back to that over and over again. And in here, there's a directory for the Comprehensive Genome Analysis Service. It has a couple of different examples of CGA jobs. One is this Candidatus raicea pediculia genome. And the other one is an assignment, but I'll walk us through a little bit of that on a Staph aureus genome. I'm going to walk us through the assembly of the Candidatus raicea genome, or the CGA of that. This genome came from the sequence read archive, so it has an SRR number. And I'm just going to copy and paste that. Like what I've done here is I've selected that CGA job just to get the SRR number here that I'm highlighting. And then I'm going to go over to up the top here, this Tools and Services tab. And I'm going to select Comprehensive Genome Analysis out of it. And that brings up the app for Comprehensive Genome Analysis. If you haven't noticed already, the apps are all sort of designed in as similar of a way as possible. So once you get the look and feel of one, you can pretty much figure out the others. We have the info up here at the top with the I button, where you can get to the tutorials and the reference guides. And then we also have the link to the stack of books is the link to the tutorials page that works you through step by step on how to submit one of these. And you can see the screenshots and stuff and kind of instructions on what to do if you get stuck after the workshop is over. There's basically two different entry points to the Comprehensive Genome Analysis Service. The first is by submitting reads. The second is by submitting assembled contigs. So obviously, when you submit assembled contigs, you bypass the assembly step. We're going to do it from reads, but that's what the radio button here at the top is. Now, you could select the reads for Candidatus raicea by going back to the workspace folder and selecting the reads that Rebecca has put there. If they are paired end reads, that can be done by clicking on that folder icon and then traversing back to the public workspaces and then the BV-BRC workshop. I'm not going to do it that way, though. I'm going to do it by adding a run accession number. And notice that this top one is for paired end reads because you have two files that go together. And the second one is for a single end read library. And then the third one with the run accession number, what it'll do is it'll go to SRA, find the reads associated with that run accession number and bring them in and figure out whether it's paired end or single end reads for you. I find that to be very convenient. Now, the next thing we have to do is we have to work through the assembly menu. I almost always choose auto. The auto assembly method is a workflow that's been worked through by people in our group, including Alan and Andrew and several others, Fong, over the years to try to provide the best possible assembly, the best bang for your buck, so to speak, for assembling reads. It is able to detect the difference between long and short reads, and it acts appropriately, whether it has long reads, short reads, or a mixture of long and short reads. So you can provide both. You can also select from the drop-down menu different assemblers, including unicycler, spades, canoe. Canoe's long reads, spades is short reads. Unicycler can do both. Meta spades, that is for assembling a metagenomic sequencing run off of short reads, although I think it could probably handle long reads as well. Plasmid spades, if you're attempting to assemble a plasmid, and MDA, which is optimized for single cell genome sequencing. So any of those are potential options for you. but we're going to select auto. And I'll talk a little bit more about what's happening on those recipes in a moment, but we're going to get the job launched first. The next thing is telling the service what it needs to know in order to do the annotation. Now, when we refer to annotation, we mean finding the features in the genome that are sort of most important to initiate your analysis of that genome. And so what are those? Those are ribosomal RNAs, RNA genes, protein encoding genes, proteins of special interest, and other features in the genome. So there are certain things that the program needs to know in order to proceed. So for instance, we have other annotation recipes, but bacteria and archaea is the only one that's exposed in CGA. And then the taxonomy name. The taxonomy name is important for two different reasons. The first is that for certain species, there are specific steps that are run by the annotation system that are not run for other species. These include finding insertion sequences in certain species, as well as the prediction of antimicrobial resistance using some of our machine learning classifiers, which are species specific. So you won't get those if you don't tell it a genus and species. Now, a lot of people ask the question, well, what if I don't know my genus and species because this is a research project? And our answer to that is that if this is a single isolate genome, try to give as close of an approximation as you can. You can select proteobacteria or gamma proteobacteria or Enterobacteriales or Enterobacteriaceae. Try to get as close as you can, and that'll result in a better annotation job. The second reason why you want to add this is because it looks up the genetic code for you. And the genetic code is important because certain organisms have different stop codons than others. So if you were annotating a mycoplasma, for instance, it has a different set of stop codons than E. coli. And so it looks those up. So I'm going to start filling in. It'll autofill for me. So Candida ricea. So I've started to autofill it, Candidatus ricea, and I think it was Platyculia, and then it drops in a taxonomy ID. If you knew your taxonomy ID, like E. coli's 833331 or, something like that, you can type it in there and it'll backfill the other way. And then a label. So when you assemble and annotate a genome using the app, it's always private. It's always private to you. However, you got to name it something so that you know what you're looking at. And this enables you to do cross comparisons in the website as well, because you'll be able to select this. So I'm going to call this UVA2 because I just assembled one this morning. And then you select an output folder from your workspace. And I have a UVA August 2022 workshop folder. And so I'm going to put it there. And then I'm going to click submit. After you've submitted your job, it will eventually show up over here on the jobs page. The link to the jobs page, again, is down here in the bottom right. And this is a job, because it's an assembly, is a job that could take a while to run. The annotation is nearly instant in a few minutes. The assembly, it depends on the complexity of the genome and what you're asking the assembler to do. And so over here, it'll list as running. It's a little bit peculiar in that it lists as running on the job scheduler before it's actually physically running your job. That's just saying that we got it. If your job fails, it'll end up coming back looking like this with a red failed status. Or if it completes, you get the green completed status. Notice also that when you submit one of these, each one of these has a job ID. And that job ID is critical for troubleshooting if you end up with a failed job. So if I go down here to my failed job, for instance, I can report an issue on that job. I don't remember what this was, but it's not important. But what it has here is this will send a ticket into our system if the job failed, and you can fill it out and say, hey, what went wrong? And you can provide a description.
Please do say, hey, what went wrong, or something about.
You got to kind of give us some clues.
Trying to figure out what.
Don't just submit it. Yeah, try to say something. So while that's cooking, I don't know if it'll finish in time because we're all hitting the website at once. There's a few things that I should show you about what's happening under the hood. So on an assembly job, the auto recipe will run Canoe with mini ASM and Racon, or unicycler if it's long plus short, or unicycler if it's short, which has a spades optimizer. Racon and Pylon are two different programs that attempt to polish, they call it polishing, the reads by doing some statistics on reads that are imperfect. I already spoke about the other ones here. When you annotate, the first thing that it does, yes?
Yeah, about the assembly options. I want to know if you consider an identify to the package.
Yeah.
Okay.
Yeah, these recipes have evolved over the last 4 or 5 years. Our original auto assembler was just spades at one time. These do periodically get updated.
Right now, nanopore reads, which are coming on strong, the system doesn't do as good a job as Fly does. When you annotate one of the sets of reads with our system, it gives you 150 context. Fly got it down to 1.
That's impressive. Meta spades probably shouldn't be exposed as an option on CGA. We have a special metagenomic binning service. And another thing I should show you, actually, let me pop out of here.
Let me just point out, Jim, because I don't know that you know this. Just for the people in this room, there's more oomph behind the assembly. So if you have something really big, even if it is metagenomics, I would do it through the assembly rather than the metagenomic binning, because the metagenomic binning will take longer because there's not as much.
Right.
There.
So there are a couple of things to unpack there. The first is that the comprehensive genome analysis service is actually 2 services in one: assembly and annotation. So you can do assembly and then annotation. So all you got to do is come over here, select the assembly service, and it's the exact same thing, only without the annotation bit. And that's what I would recommend for a metagenomic assembly, just if you just want to assemble. The second is obviously annotation. Now, annotation is a little bit different because you can do a simple annotation run. It'll look the same, but CGA gives you a little bit nicer of output, and the actual underlying annotations are identical. So I'd probably do an annotation run when you already have your assembled contigs from the annotation service. And then if you're seeking to find the nearly complete genomes within your metagenome that were assembled, we have a binning service that does that. And that's next. So I don't want to be a spoiler to that. So the underlying annotation system is RAST. Has anyone ever used RAST from the RAST website? So it's doing almost the exact same thing. There's a couple of really minor differences, but basically under the hood, this is RAST. And I'll go through the steps here. So the first thing that the RAST pipeline does is it looks for ribosomal RNAs. And those are sort of the first essential thing you got to find in a genome. They're not going to have other stuff overlapping them. You just want to get those called and out of the way. The next thing it does is it looks for tRNAs with tRNA scan, and then it searches for repeat regions. These are all non-protein coding bits. It searches for repeat regions literally by doing a blast against itself, and it calls that a special feature in the genome. The next thing it does is it attempts to find special proteins. We sort of throw the term special proteins around a couple different ways, but in this respect, we have our own special software that looks for selenoproteins and pyrolysyl proteins. Selenoproteins and pyrolysyl proteins are special because they contain a stop codon. And there's a mechanism by which a selenocysteine or a pyrolysine gets placed into that amino acid sequence. So what happens when a traditional gene caller is scanning the genome is that it interrupts the open reading frame. And so you end up with 2 pieces of an open reading frame instead of 1 full open reading frame. So that's why we have a bespoke tool for finding those. The next thing it does is it looks for CRISPRs. These are clustered regularly interspaced short palindromic repeats, and everybody knows CRISPRs at this point. The next thing it does finally is it gets to the point where it's calling open reading frames, or the protein encoding genes specifically. And this is done using both Prodigal and Glimmer. So if you start comparing your GenBank genomes to our genomes, this is where most of the differences are, because the Prodigal and Glimmer gene calls are essentially predictions that are based off of hidden Markov models, though I believe that's how Glimmer and Prodigal work. And there's a little bit of wiggle room in how those protein encoding genes are called, because you have the potential for multiple start sites, and there's just noise. Glimmer has a tendency to call shorter protein encoding genes, in my experience, than Prodigal. Kind of like Prodigal a little better than Glimmer. But at the end of the day, what we do, and I'll talk about this a little bit later, is that we go through and we merge the results, and that gives a cleaner set of open reading frames. Then we assign function. This is the special sauce that makes RAST different than every other annotation system. It works by having a set of signature k-mers. These are amino acid 8-mers that are unique to the specific protein functions that have been curated in the seed database. The seed database being a database of protein functions. In the world of RAST, what our annotators try to do is they try to assign one or more atomic protein functions to a protein itself. Those are called functional roles. And so you can have a protein with more than one of these, but essentially those are what are being annotated onto your protein encoding gene whenever it gets run through the annotation system. And this is done through this hashing, these signature k-mers that are signatures of specific functions. So the first attempt that it does is it blasts against a database called the core seed. That's where all of our active curation is happening right now. And the proteins that are annotated against the core seed are usually very highly accurate and consistent. And then the second attempt is that it will blast against close relatives. So if there happens to be a function that we got through GenBank or some other means, it will inherit that from its close relatives. Then finally, the next step is that it will take all of the open reading frame calls and resolve any overlaps. And this is sort of an optimization scheme that attempts to place all of your proteins, if it was a circle, around the genome without having very many overlaps, unless they're absolutely required. It's not perfect, but no system for resolving these is, which is where the research still is, obviously. And I should note, since we're talking about resolving overlaps, is that there's a special annotation recipe for phages because phages are highly overlapping. So if you're annotating a phage, you should choose the phage annotation recipe on the annotation app and not use comprehensive genome analysis service. Now, after we have all those protein encoding genes and all of the RAST functions and everything applied, there's still more things that we want to look at as users of BV-BRC, which include some of our other proteins. We call these specialty proteins, which they're basically blast matches back to curated databases of important things. So CARD, if you're studying antibiotic resistance, CARD is a well-known resource that contains antimicrobial resistance proteins. NDARO is NCBI's well-curated antibiotic gene database. Now, there's a little bit of magic here in that our system is entirely protein-based when we get to this point, but the NCBI's system is gene-based. And so when you see a match to the NCBI set, it will be at the protein level, and that will also be reflected in its annotation. And so in the aminoglycoside resistant genes and in the beta-lactamase resistant genes specifically, the nomenclature of those goes the whole way down to the nucleotide level. And we capture that in the annotation of the protein. So you'll see these very sort of long and Baroque protein annotations for those, but they cover the phylogenetic unit that's represented by the small SNP differences in the beta-lactamases and the aminoglycoside resistance genes.
So if I'm understanding correctly, your annotation pipeline doesn't use a VED reference genome?
No. Well, there's nuance. There's nuance. If you gave us an identical genome to one we did previously, it will inherit the annotation faster that way. I think it still does that, doesn't it?
No.
No, it used to.
RAST does that.
Okay.
So like I work with E. coli, right? And if I were to use PROCA to annotate an unknown clinical sample and I fed it MG 1655, that wouldn't be great, right?
Right.
But with this, I don't have to.
It's de novo. It's de novo. Any protein that we have curated, it will call. As long as you get an open reading frame for it, it will assign that function. It's not inheriting it from another genome per se. It's inheriting its functions from the entire collection. Does that answer your question?
Yeah.
Okay. There's obviously pros and cons of that, right? If you had something that was nearly clonal and that you had been curating, you'd want to inherit those. But if you have something from the environment or something that you've isolated, this is probably the better approach.
And if that gene is particularly specific to that organism, then it's difficult to find de novo, right?
Not if it's been annotated. If it's been annotated, we'll find it. If it's not been annotated, it'll come up as a hypothetical or something like that.
Things are
Yeah,
hypothetical with the potential assigned function, right?
no, we haven't been doing that. Usually when we give you a hypothetical, it just says hypothetical protein. If you see a function that's hypothetical and then it has a string after it of characters, those are almost always from some other annotation system that have been inherited or legacy, it's still in the system somewhere.
Hypothetical means it has some homology with other.
They can. So this is where we get out to the limit of what the scalability of a human annotator. So our humans go through and they assign functions to proteins in the seed, and then that gets propagated out onto all of the BV-BRC through the annotation system. But there are things out in the fringes that they haven't gotten to. I mean, our core strengths are metabolism, antimicrobial resistance, transporters, good core stuff. If you're studying things that are like way out in the pan genome that only occur in 5 or 10 organisms in the database, they're probably going to come up as hypotheticals. However, if you have good literature references and good examples of proteins and you know what they are, you can submit those as a ticket into the system and say, hey, I know exactly what this is. Here's the papers for it. This has been characterized. Can you add it to the seed? And we'll send it over to the annotators to get it added. Now, the update process can take a while. It could take 6 months to get it out into the annotation system of BRC, but at least you're getting it in, right? There aren't that many groups left that are doing annotation like this. So it's one avenue in which you can get that into the public hands.
Yeah, so I think the people who are working with this kind of organisms that are not well covered or not similar to kind of like some of the other pathogens that we have in the system, we would be interested in knowing what those pathogens are. And maybe during the break or something, we can look at some of the public genomes that are there in the system and see how well those genomes are annotated. And if you happen to know more about those organisms, then you can probably take a look at it and tell us whether you think we have enough coverage in terms of assigned function, or whether there is a major gap between what we have and whatever are other favorite tools or
Right.
databases, what you are getting from that. If that's the case, then I think we can systematically go after them and try to improve the coverage.
Right. These specialty protein databases that we match against, they serve as like a safety net for a researcher who is familiar with a different nomenclature or is worried that they might miss something on our side.
So we are often surprised because in all of these workshops, there are always people who are working with non-pathogens. And even for them, the RAST and the BV-BRC annotation system has been working wonderfully. The only problem, I remember one time we went to Germany, and most of the people it was a marine microbiology lab, and they were collecting a lot of these deep sea organisms. And they were complaining that there are significant number of portion of genes which are assigned as hypothetical compared to some of the other pathogens. And when we investigated it further, we found that it is because there is nobody else out there. Nobody has tried to characterize the function of those genes, right? So if there was some knowledge, some literature, some experiments that were out there that try to expand the knowledge with respect to this organism, then I think we can improve.
Right.
State of the art for this organism is that nobody knows what those genes are. Yeah. So if that's the case, it's a completely different story.
Yeah, and I'm jumping ahead of myself here, but there's also, as a final fallback, we assign protein families membership to each protein encoding gene, well, to each protein sequence. And those are Kamer-based and homology-based. They'll be blast-based, so they're homology-based if they're hypothetical proteins. And so you can still get clustering of related proteins, even if they're all called hypothetical. So that's also of some use, especially if you're a person who's trying to elucidate the function of a particular protein, then that should be of some use. Some of the other specialty databases we match against are for virulence factors and drug bank. Those are drug targets, so is TTD, and then also human homologs for people who are looking to develop out pharmaceuticals and stuff. It has an algorithm for finding close neighbor genomes, and then it does AMR prediction. The AMR prediction should be explained. This is an ongoing research and development area of interest in our group, and it's of particular interest to me. Right now, we have a set of machine learning classifiers that will predict susceptibility or resistance to a variety of antibiotics for a select set of well-studied organisms. And those include strep and staph, salmonella, mycobacterium, a few others. The production system has an earlier version of our classifiers. It runs off of AdaBoost. And they're pretty trustworthy classifiers. It's a k-mer-based machine learning algorithm. So what it does is it finds the k-mers that are indicative of resistance only, and it maps those onto the genome as a feature. If you're unfamiliar with machine learning classifiers, they're like essentially fancy weighted voting schemes. So the k-mers basically vote as to whether or not they think it's a susceptible or resistant genome. And so you can actually browse those, which I'll show you in a moment. So that's what's happening. And that's one of the reasons why you want to tell us the genus and species name. So if you're like me and you run a zillion jobs, come back here, look at your job status, it's still running. That's unfortunate, but we do these workshops like Julia Child used to do the cooking shows. So we have a completed one in the oven that we can pull out and show you and say, voila, look how good this is. So here's one I did this morning. It's the next one down on my job list. This was the first one. So there's a couple different ways that you can access a completed job. One way is through the jobs link down here in the bottom right. You can always click on that and that brings you to the jobs page and then you pick the job that you care about. Another way is that workspace folder that you selected to have your job results in. You can always navigate to that. So I can go to my home directory and I can go over here to BV-BRC workshop, and I call this UVA. And so this is that particular job that I ran. It's the same thing. When a job completes in your job directory, you always get that checkered flag icon, and you can click directly on that icon and it opens up. It is a directory unto itself with all of your job results. And since this is the comprehensive genome analysis service, there's a lot of job results because it probably does more than any of the other services, except maybe metagenomic binning. Any of the ones that are listed as HTML in your job output directory, you can actually view natively. So I can double click that and start walking through the output of the comprehensive genome analysis service. And we tried to model this off of, like, you know how people make these papers on single genomes, these genome report papers. We tried to kind of model the output on this so that it would be easy to sort of pick up the pieces and try to do that if you wanted to do that. So the first thing is a set of general statistics on the assembly itself. Looks like we had 6 contigs with a GC content of 28%, which is pretty common for an endosymbiont or a parasite. Genome length, number of plasmids comes out as 0, and 50, that's the length of the contig at which, if you count backwards, you reach half of the genome. Chromosomes is not listed because that's covered by contigs in this case. We have the job ID, when it started, when it completed, and total time it took. So this particular job for me, back before everybody was hitting the website, took 8 minutes. So, but this, remember, this is a tiny genome, for example. The next thing is that it starts to run down some of your annotation results. So it gives you number of CDSs, number of tRNAs found, number of ribosomal RNAs found, et cetera. And we got the start and stop and the actual annotation job number. And then the next thing is that it starts talking about the functions. So we attach EC numbers, that's enzyme commission numbers when we can. We attach Go terms when we can, number of hypothetical proteins. If we have an EC number, we can assign it to a pathway. So metabolic pathways, and there's a viewer for those that I can show you. Number of protein families that it gets. In this case, there are 2 different levels of protein family generation in Patrick right now. The first is global and the second is local. The global families will try to aggregate as many proteins into a protein family as possible. The local ones will only build families on a genus. I actually prefer to work with the local ones, but because this is a candidatus, it didn't do that. So that's why you get a 0 there. The next thing is the circular genome viewer, AMR genes, CDS with homology to known virulence factors, GC content, GC skews. So you guys can go through that. Now, the Circos plot, this is generated by a program called Circos. And if you remember when we were scrolling through, this thing has 6 contigs. It orders them from longest to shortest, and it makes them a circle by default. So this is not necessarily a circular genome. It probably is. But look how, like, it kind of cheats a little bit, because if you look up here, here's your first contig, second, third, fourth, fifth, or maybe there's 2 here. Oh, there's 2 here. So it makes it look a lot more beautiful and perfect than it is. And in reality, we don't know what combination those come in, because if the assembler couldn't figure it out, then more work would need to be done to build scaffolds or something in order to figure that out. Just word to the wise. It looks great, but don't trust it. I mean, you can trust the assignment of coding sequences and stuff to it and all of that. You just can't trust what order they're in. or that necessarily that it's circular, although most bacteria are. The next is the subsystem analysis. Subsystems are groups of proteins that perform a related function. So for instance, if you had a subsystem called transcription, it might have RPOB or RPOC, RPOA, like the bits and pieces of the RNA polymerase. And so this is a breakdown of the various subsystems. And so people find this useful as a first approximation of what's there. Specialty genes we spoke about at length, number of matches to card, Patrick, drug bank, TTD, TCDV. Now, this match to card is based off of homology. So that's probably a match to an RNA polymerase or ribosomal protein in card. Just because it matched card doesn't mean that it has the exact SNP from the exact organism that it has to match in order to be resistant. So this might or might not be resistant. Like more work would need to be done in order to align that and make sure that the SNP is there in order to impute resistance from the sequence itself. And then this is the list of AMR genes that got found by those different things. And if you look down through here, we got RPOB, RPOC. You see what I mean? You got to look at those with the prior knowledge that it will find them based off protein similarity. It also does a rudimentary phylogenetic analysis for you. I don't know that I would necessarily pick this up and drop it into a paper. However, it does give you a lot of information fast. So it tells you a lot about your Candidatus raicea genome because it shows you where it lives on the tree. It's down here. You know, I don't know offhand how these others are chosen, but here it is. Minhash.
So it's just pointing you in the right direction.
Yeah, it's pointing you in the right direction, but I'm kind of looking at this one a little skeptically. And then down here's a list of different references for the various tools that were used along the way. That is what the HTML of the full genome report looks like. If you go back, that we got our breadcrumbs across the top here. If we follow that back to the output of the CGA, because the Comprehensive Genome Analysis Service is a mashup of the Assembly Service and the Annotation Service, there's actually two more job directories baked into this directory. So there's the annotation and the assembly output. So there is a special assembly report that you can look at. And so what I did is I clicked on the assembly output and then I clicked on the assembly report. I'll go back and do that again because I did that too fast. So here, that's my output for the Candidatus raicea CGA job. And then I clicked on the flag for assembly. And now I'm clicking on my assembly HTML. This is the bandage plot. Bandage plots are really cool because they show you where the assembler couldn't get the job done, where you have a problem in your De Bruijn graph. So you can kind of see where you had repeat regions or whatever that caused this to break down. In the grand scheme of things, that's a pretty good bandage plot. They don't get that much better unless you're doing like combinations of long and short reads. This gives you a breakdown of the assembler parameters and version. We did unicycler 4.8 and then other things like that. Average depth is 600, so that's pretty good. Minimum coverage threshold was set to 5. Minimum contact length displayed is 300. So you get all the data that you need out of here without risking boring you. You can go the whole way down through here and get all these stats on your assembler itself.
Very often when I run assemblies to Patrick or BV-BRC, I'll get bottom of my bandage plot, there's kind of little orphan chunks of sequence at the bottom. And I wonder what those represent.
They represent ones that can't be put together, I think.
Okay, might be plasmids.
Could be plasmids, yeah.
That would be excellent.
Yeah.
If you look at the contents that came out, you'll probably see a bunch of probability content down here.
I think that the qualitative characteristics are important to look at because every assembly is a hypothesis anyway, right?
Right.
And so imagine I wrote a really bad assembly algorithm that just concatenated all your reads together. I just joined them all using ends. I give you one config every time, right? No problem.
Yeah, and you feel really good about yourself without being
Right.
I want to know what defines the presence or absence of a plasmid, because you could extract a pure plasmid or encode a pure plasmid and try to assemble it or do annotation. And I see my bandage plots almost closing as circular, but it still reports to me that no plasmid. I know that I'm going to see you guys.
This is a really important point, and it's
When I know that I'm dealing with a plasmid.
right. To repeat what Alan said, the unicycler report in the FASTA header will say that it's likely circular, and most plasmids are circular. Or you could attempt to assemble with plasmid spades. And sometimes the depth of coverage is much greater than that of the surrounding chromosomal regions. However, the BV-BRC currently does not contain a tool that says, yes, this contig is a plasmid and this contig is a chromosome. There's lots of ways that you can kind of figure that out as a researcher by going through and looking for the presence of universal genes or replication machinery if it's a plasmid, or conjugation machinery. But right now, we do not offer a prediction as to whether or not a contig is a chromosome or a plasmid. Now, I think that we're on the brink of changing that. I think within the next 6 months, potentially, we have a clean slate of projects that we could probably get. We have 3, potentially 4 papers on this topic, building the various machine learning algorithms for trying to predict whether chromosomal sequences are plasmids or contigs. So I think we should have tooling for this eventually, but that's sort of right at the cusp of research transitioning over into the system right now. It's something people have asked for years, and it turns out to be kind of a tricky problem, machine learning wise, because plasmids are all different lengths, but tend to be small, and chromosomes as a contig could be all different lengths, but tend to be large. So there's a little bit of work that has to be done to make that all work out. Okay, so he's asking how we estimate coverage relative, because it's a de novo assembly. And I believe that the assembler does that, but I could be wrong.
Essentially maps the read back to the.
We use SAM tools to map the reads back.
Oh, we do? Okay, there you go. So the answer is that we use SAM tools. I thought that the assembler itself offered an estimation.
Some do.
Maybe that's why we use SAM tools to go back.
Open the assemble contig file, maybe contigs.txt, I believe, in the assembly. Download and open it.
There it goes.
Okay, so if you look at the faster definition line for each of the contig, you will see the length of the contig, you will see the coverage for that particular contig. So it is basically doing the weighted average across all the contigs, right? And then that is the equivalent number that you'll be getting for the overall coverage for your.
Okay. The rudimentary tree file, the rudimentary Circos plot, all of those things exist as SVGs that you can download and turn into high resolution images that you could work with later. Obviously, for the assembly and the annotation has the FASTA file of your features, which are your protein encoding genes and your RNA encoding genes and everything else. The FASTA file of your proteins, GenBank formatted files, GFF. There's tables of the annotation. There's an Excel sheet with the annotation data. And so there's a whole bunch of various formatted files that you can look at. So for instance, I'll grab the annotations because they're the most fun. So here is literally all of your annotations in an Excel file. You have a contig, you have a feature ID. You get used to seeing those throughout the website, what type it is, whether it's RNA, CDS, or some other predicted region. Start and stop, what is it? Large subunit rRNA pseudouridine synthase. PGF is the protein family ID, nucleotide sequence, amino acid sequence. So there's all kinds of different ways you can work with the data, and that's just one of them. The various formatted files of the output only get you so far. What's really most interesting, and perhaps the most interesting aspect of the annotation service specifically, and the annotation service as a subservice of the CGA, is that your genome gets indexed against our database. So you have a private genome now that's in our database, and now you can start looking at it from the website. So I'm going to show that again. Whenever there's something that you can view on the website, there's this button up here in the top right corner called View. It's an eyeball icon. And that enables you to go to the website and actually look at the genome that you just annotated. It contains a lot of the information that you have seen already. The format's obviously different, but it loads the various features of your genome, how many genes you had. Here's your taxonomy ID. Here are the tables that we're familiar with from the output already, the protein features, hypothetical proteins, drug targets, and so forth. And except now, the links are live. So if you wanted to study your hypothetical proteins, you could just click on the button. There's 79 of them, and it'll load them up into a table for you. And then you can download those specific proteins and go on and do other things with them. So here's all of our hypotheticals, for instance. If I go back to the overview tab, and I wanted the 16S, I could click here, and there's all this different data about the genome on the landing page that can be explored. But the important thing is that now it's in the context of the other stuff. There's an AMR phenotypes tab. I'm going to show you that for a different genome because this one isn't really resistant to anything. There's a phylogeny tab.
That is for the order that your genome is in.
So that your genome does not show up as a tip.
No, because it
Okay.
took us months to run those things. There's no way we could.
Okay. The genome browser. This is one of my favorite tools. The genome browser is based off of JBrowse, and it enables you to walk the contigs of your genome. So each one of these is a feature that's been called in your genome. And it does this by default. For one of the genomes in BV-BRC, it'll have a RefSeq annotation track if we brought it in from the public. This one didn't come from RefSeq, so I'm just going to blow that away. But you can zoom in and out and do mouse overs of the various protein encoding genes and see what they're annotated as. If you zoom in, it gets sort of bigger and easier to see. Up here, you can scroll through your 6 different contigs and select each contig to look at it. There are link backs to this from each feature page. So if you wanted to look at an individual feature, this is ATP-dependent protease LA. You can go to the page for that particular feature. It tells you what contig it's on. And you can zoom in even further. So if you're really interested in looking at the various SNPs, you can get the whole way down to the nucleotide level and look at your open reading frames. If you didn't believe your start positions, for instance, which are probably the most frequently broken. So here's one, oh, these overlap, the stop of this and the start of this overlap. I wonder if that's the truth or if that's something weird. I mean, it looks like an operon to me, cytochrome O ubiquinol oxidase. Cytochrome O ubiquinol oxidase subunit 3, and this one's subunit 1. Yeah, I wonder if 2 is in there somewhere, So it's that sort of thing that this is really particularly useful. The circular viewer is just, it starts out as the original one from the CGA output, but you can actually change the tracks on here, and that'll get demonstrated later. So you can generate one that suits your needs, which is pretty nice. Sequences, we're getting into some of the dirt on the website right now. Sequences means contigs. And there's just this ancient, I don't know, reason for it being called sequences. Okay, so here's each of the 6 contigs, the genome identifier. There's your 6 contigs. It'll show you the GC content and the length. If you wanted to work with one individually or download it, you can select it. You can look at it, go back to the genome browser and look at it. Down here, you can get the FASTA file for that particular
That's the
contig.
only place that you can grab that.
That's the only place you can get a single contig,
Right.
right? And it's under sequences, but it should be called contigs. The next one is worse, probably. This is a new screw up on our part. This is the features tab, but it's called proteins because
Yeah, naming is the most difficult part because trying to get a consensus on what to call something, this is like naming your baby.
But this one's bad.
Everybody
This one's
has strong
particularly
opinion.
bad. So it comes out pre-filtered, which is probably not useful to us, but there are different features depending on the genome that you have. So in this case, we have the option of all of them, coding sequences, protein encoding genes, ribosomal RNAs, tRNAs, and whether
The private genome.
yeah, so so it comes out false. So basically, if I would just wanted protein encoding genes, I could select CDS, and that's from this filter tab. It looks like a funnel up here. If you had AMR classified regions, repeat regions, some of those other things, they'll show up in here as well on a bigger genome. Now, protein structures, will this load for this genome?
No, because it's private.
Ah, I thought that it did a match.
No, we don't have any.
No,
Specialty genes. These are our matches to CARD and NDARO and the others. So you can look at those individually. Really useful on an AMR, on a genome that's resistant to something. Here on the specialty genes, you can see it's actually BLAT and not BLAST for most of them. Or if it's from Patrick, it'll be based on a camera search and it'll tell you what the various genes are. But in a lot of these cases, they're matches at the protein level to EFTU. You can see how many EFTUs are there, S12P, S10P, those sorts of proteins.
now I designed these things to make it easier for you. Scroll down to the bottom, please. And he did mention this before. These are all the references for your particular genome. So when it comes time to publish your paper, remember, there's a lot of path to publication that I've baked into this, right? So I've got those here for you, so you don't have to try to figure out exactly what version of the assembler we used and what algorithms we use. It's there for you, and in fact, there are places where you can just cut and paste. The way I was envisioning it is to take it for your materials and methods section and just dump it into that. And then, of course, with EndNote or whatever, you're going to have to put in those references. But we want you guys to publish because we want to make you happy. And when you publish, it makes us happy and it makes our program officer happy. So all we ask, it's a free service. We just want you to cite us and reference us. That's all you have to do. And all of this is available for you.
There's one more thing I need to show you. Sorry, Rebecca.
Uh-oh.
I ran
Oh,
in
yeah.
the assignment, there's a staff genome, and I did a foolish thing where I assembled all the reads separately, and it was like a combined long and short read assembly. So you can try doing that. If you look at that particular genome, one of the reasons that we wanted to show it is because it actually gets AMR phenotype calls. And remember, these are predictions. They're not always perfect, but I wanted just to show you what that tab looks like when it actually does get populated. If it is a genome that is one that you submit that is of a type where you can get an AMR prediction, you'll get your genome ID, the antibiotic, and whether we believe it to be susceptible or resistant to whatever antibiotic, and then what the evidence is based on. And right now, these are the AdaBoost classifiers that are in the production system. If it's one from a public genome, and we've been curating public genomes out of the literature for years now with AMR data, these fields in the middle, the measurement sign, the measurement volume, the measurement unit, and the lab typing method, they'll most often be filled out. So you can tell the difference between them because it'll either fill out as a computational method, which means it's predicted, or it'll fill out as an actual measurement sign from an antimicrobial susceptibility test of some type. And the type of test will be listed. And if it has an MIC, you'll get an MIC. If it's got a disk diffusion diameter, you'll get the diameter, or you'll get SIR, and whether it's based off of CLSI or UCAST or any of those things. There's also a way to download those from the FTP site, which I can show you later. But there's about 67,000 genomes in the database right now that we've brought in with AMR phenotype data linked to them.
These are just predictions.
That's just a prediction, right? Okay.
Okay,